Okay. Well, yeah, let's just wait 1 more minute. Maybe a bus is just arriving. Okay, so this is week 12. Any questions with your homework, the last homework? Open question. Homework is no right or wrong answer. I just need to design, come up with an idea, right? Okay, let's go ahead and start today. So we are going to discuss. Yeah, good.
I'm sorry for the delay.
No.
You said we should read that paper and apply to a new biological question.
Yeah.
So the question, could it be something that maybe like they had used the kind of like error-prone method to do their analysis, but maybe you apply it to a new, like a previous data set that has been worked with already, but with the old approach.
You can, yeah.
You apply this new approach to that data set.
Mm-hmm.
Is that okay?
A new approach.
Like this approach from this paper
Yeah.
to data that has been already analyzed with the error-prone method. You are applying this approach to that data set.
It's which data set is not very important.
Okay.
The method you tweak. I want you to find a way to tweak existing algorithm or method, and to an angle which people have never thought to use this method to that problem. So you can imagine and this new application, new way of application of existing algorithm to any algorithm, any data set. It doesn't matter which data set it's about, as long as your idea is there, you know. Nowadays, if you have idea, you know where to go, and everything else, AI can help you. But I don't know how much AI can help you to have this new idea. If you have a smart AI can help you generate a new idea, it's okay. You can apply. Yeah. Okay, so manifold and tensor. So there's two concepts which I found useful to understand what they are, and you can find a lot of applications in different occasions in single-cell data analysis, and also other high-dimensional data analysis as well. So I found it's important to know this and to open your mind to see, okay, we do have those technique available, and when you have enough different batteries of algorithms, then you can weaponize yourself, right, to have more options. So we will see manifold learning as a nonlinear dimensionality reduction technique. And I'm going to use two examples to show you how you consider the problem to solve this nonlinear dimensionality reduction. And T-SNE UMAP are one of these. I'll give you two more to have your get better understanding of those applications. Then we'll mention if you have the manifold concept, we can do manifold alignment. You remember last week we talked about the CCA, canonical correlation analysis, to align two data sets to make them correlation coefficient between them after embedding as much as possible. This manifold alignment is a nonlinear version of that, basically. So how can you align two manifolds? Then there are some applications, and I will mention this multi-omics data analysis using single-cell RNA-seq plus single-cell ATAC-seq as an example, so you know what the problem we have if the same measurement from same cells or not from same cells, what we can do. Be a tensor, which is a high-dimensional matrix, and correspondingly you have a tensor decomposition method and some applications example. At the very end, we will have a very quick introduction of this RNA velocity analysis. So manifold is a term that's mathematical term, is a topological space in which you have this nonlinear structure of representation of your data points. So if you look at this structure, if you are, this is a 3D object and very complex. If you are a 2D animal, you are an ant, you are not a grasshopper, so you will be put on this surface. If you want to move to here, you cannot jump. You have to go through this structure, okay, in order to move from one position to another. Sometimes these two positions maybe look like very close to each other, Euclidean distance, but you cannot jump because you are 2D animal. You cannot jump from here to here directly. You have to go down, and the move may take a long way back to here. Okay, so that's what I mean. So locally, your Euclidean distance makes sense to you because you move here to here. You can compute in this local distance, but you cannot apply same rule when you compute non-local. So from here to here is non-local. You cannot just build a straight line from here to here directly. You have to consider the whole structure of this complex topological object and follow the structure of this topological object. But most of the time, this is abstract mathematical objects. It's not real, but you have an imagination in this high-dimensional space. You can have those. So I will mention this locally Euclidean concept again and again because that's the whole point for this. I will mention this distance looks like close to each other, but in reality, you cannot move, you cannot jump. Okay, so when you deal with this manifold structure, and you need to apply dimensionality reduction technique in order, for example, just visualize the relationship between data points. You need to have this nonlinear thinking. So consider using nonlinear dimensionality reduction to the manifold structure. So this process is called manifold learning. So manifold learning is a subfield in the group of technology in the nonlinear dimensionality reduction. So manifold learning is one of the major one, basically. So it's very different from a PCA. PCA you consider as a nonlinear dimensionality reduction technique. Okay, you consider the whole thing. You consider this whole object without considering in the local details. You just rotate and project, look at the shadow, but you don't worry about this very complex structure within this manifold. Oh, it's a simple fragment, nonlinear structure. So dimensionality anomaly is called unsupervised learning, and you just look at the data structure itself and to perform without any training data. Just like PCA, right? Okay, so this is another illustration. See what's called local Euclidean. So this is 3D and shape, okay, S shape. So if you look at this local patch, and you can compute the distance from one position to another, okay, just use their X, Y, Z coordinates. It makes sense. You move from here to here and follow the Euclidean distance. But if you compute a distance, for example, from here to here, okay, in that case, you cannot use Euclidean distance because this structure is a manifold structure. And if you want to do this dimensionality reduction from a big D, the 3 to 2D, small d, okay, you do this dimensionality reduction to project your data points in X into Y. So this is dimensionality reduction. You want to keep not only this local distance relationship, but also between all the data points, okay? So you want to do something like this. If you illustrate with this 3D S shape, you want to unwrap this to spread it out. So the distance between this to the center, for example. Okay, you after you spread it out, you have to go from here to the center. You have to go through those yellow, orange points, okay? Because the structure should, you have to go through this yellow, orange until you reach out this dark red area. You cannot jump from blue directly to dark red. So this is a good dimensionality reduction which reflect the real relationship in the high dimensional space, their distance, okay, global distance. So that's what I mean by this manifold learning to do this nonlinear dimensionality reduction. Okay, so as I said, manifold learning algorithms, there are many of them. So we will using isomap and Laplacian eigenmap as an example. There are many others. They all belong to nonlinear dimensionality reduction. So manifold learning is one of this important subset of nonlinear dimensionality reduction. I think in the Wikipedia page, if I click, I think that's a mistake. It says nonlinear dimensionality reduction, also known as manifold learning. So So this definition just make two concepts equal to each other, but I believe this manifold learning is only a subset of this nonlinear dimensionality reduction technique, because some of those are not manifold learning, autoencoders, for example. But a lot they are. For example, diffusion maps, LLE, this is all manifold learning. Okay?
So manifold learning is a type of nonlinear dimensionality reduction.
What's an example for this? For example, nonlinear PCA, I believe this not belong to manifold learning algorithm. But yeah. Autoencoders, I don't believe this is. This is some neural network structure algorithm. T-SNE, and this is T-SNE, right? This is a manifold learning algorithm. There are some features in those manifold learning algorithms which maybe you figured out once you have the examples to show you what that means. So basically, you need to have ideas to consider your data as a manifold. Your data form data points and construct or form a manifold structure. And you try to model or try to compute this local neighborhood structure, okay, from your data. If your algorithm utilize this feature, utilize this step, and you have this modeling, manifold modeling, and your algorithm belong to manifold learning. So how you found this local neighborhood structure? The feature, again, is locally Euclidean, and the small distance you will compute using the small steps. But for the long range distance, then you cannot do this. You have to, you cannot directly compute the long range distance directly. You have to build through this accumulation of a series of local steps throughout your data points. Yeah, so we normally believe this cellular space should be modeled as a manifold. It surprised me to me that those very simple, so for example, this isomap algorithm has been published or proposed very late in the year 2000. So that's 26 years ago only. But if you consider how simple this algorithm is, you will feel surprised. So this is a science paper, by the way. It's global geometric framework for the nonlinear dimensionality reduction. So that's figure 3, have a Swiss roll data set. Okay, that's your distribution of your data in the 3D, have this shape. So the example is between these two data points, you cannot compute and utilize Euclidean distance directly. You have to follow the structure of this manifold and find the distance between these two data points. So ideally, if you have a good nonlinear dimensionality reduction, you reduce this 3D structure into 2D, okay? The distance between these 2 points should not be very close to each other, should be spread out. That's what the ideal situation is. From here, you move to this. You have to follow this long trajectory and move almost the whole structure of the this object. Okay, so that's the idea. So how this ISO map algorithm is designed. So it's based on MDS. I hope you still remember what's MDS. That's a multi-dimensional scaling, right? So you start from the distance matrix, pairwise distance matrix, okay? My example was in the lecture 3, and it's a city, okay? You have a map, and you want to use the pairwise distance between cities to reconstruct this map. So that's the idea, the foundation for the ISO map. The only difference is the distance we use to compute is geodesic distance rather than the Euclidean distance. So once you compute the geodesic distance, the rest of this procedure is the same as a classical MDS. Classical MDS to compute the distance between data points. Then you obtain a pairwise distance matrix, the squared matrix, and then MDS will project your data points into a 2D map, and you know, okay, the distance follow that. So now you replace the values in this distance matrix using the geodesic distance to replace classical Euclidean distance. That's all. So this is one way is to compute the short distance parts between two nodes through this Dijkstra algorithm that's compute the shortest distance in the network. Okay. So I will show you this will be based on a KNN network, and then compute the shortest distance by travel through this network and found the so this is graph theory algorithms. And if you read this, the top n eigen vectors of geodesic distance matrix represent the coordinates in the new n-dimensional Euclidean distance space, Euclidean space. So if you cross out this geodesic and using Euclidean distance matrix, the whole thing here is the last step of MDS. Okay? So all representation here, the description is what MDS is doing at the last step. So that's what I said. This is a very simple algorithm, but it's developed very late, proposed officially. So you're just really using geodesic distance to replace Euclidean distance, and everything else are the same with MDS. So what is geodesic distance? So this is a map, and 3D map, basically. So you cannot travel from this end to here directly through this dashed line. You have to move, follow the landscape, and the right curve is a real distance. It's a geodesic distance. You follow this landscape, the real distance. If you have a river, you have to go through the river somehow. So after you embed this reduced dimension to 2D, you have to respect this distance, follow the geodesic distance, which makes this 2 points distance almost the same as this right curve rather than shortcut, okay? same as here, so you cannot calculate C and A and B. They are very close to each other, but once you project them to the low-dimensional 1D space, follow geodesic distance A and B has to be far away to each other rather than collapse onto each other if you used Euclidean distance to do this dimensionality reduction. So that's what we know about the geodesic distance. And this is one of the applications in the neuroscience. They collect FC, this is called a functional connectivity map. It should be some neuroscience measurement in the brain area. Maybe use functional MRI, I don't know exactly, but you collect the samples from different locations in the patient 1A, for example. And then you have data from patient B. Now you have a new data set comes in. This is sample X. Then you want to, through computation of the distance, to predict or assign whether this participant X is A or B, okay? So from the description of this paper, we know if you do Euclidean distance, then the distance between A and B is smaller, okay? Smaller than A. So if you use, for example, Pearson distance, then you will assign X to B sample. So only when you use geodesic distance, then you will assign this correctly to A. The existence of this manifold structure. So if you follow the geodesic distance, you know X is much closer to A compared to B. So this is a real identity. So the researchers know this is this data is from A, and then they confirmed you have to use geodesic, so-called geometry aware distance in order to compute. So this is one of the applications for manifold structure in the neuroscience. Another is illustration. So from A to B, you have to move through this to get geodesic distance to replace your pairwise distance matrix in order to do the correct dimensionality reduction. So how this algorithm is implemented or compute. So we need to find this k nearest neighbor network, and then once you have this network, you will find the shortest path between any given two nodes. So you have so many points in your data. You basically have to compute all the pairwise distance between them, but follow this network structure. The algorithm you can use this. There are two options, and there are several more, of course. Then the last one will be MDS. Okay. So K nearest neighbor graph, so KN graph, is one of the, I would say, most important way to identify this local structure or modeling data as a manifold. You have this KN graph to guide you to move through the local neighborhood. So if you want to travel to the long distance neighbors, you have to follow this KN structure, move small steps in the graph. So you need to create this KN network first for all the data, the points under consideration, then move through this network. So if you have no idea what KN is, we should have a previous slides have this. Remember, maybe this was last time or previous weeks. K equals 3, K equals 4, create this more connected KN network graph. So that's one way. Another way is called data diffusion, okay? So in the data diffusion, you will create this pairwise distance matrix. And this matrix contains all pairwise distance from any given point. You can find its distance to all other points, right? Then you apply a filter. See, I need to remove the link between this node, this data point, to any other data points, which the distance above certain threshold. So you set up a cutoff. So this cutoff will trim to cut those long distance linkers, okay? So if you have just examples, those points, I can only draw 2D, but it could be a high dimensional, right? So if we use first method, it's KN, so you just link, you only link the nearest neighbor. So you just do this. Okay, something like that. You make a network directly. Just found the K nearest neighbor and make a link. Well, so when you travel from this point to here, you have to go through 1, 2, 3, 4. That's 4 steps. That's the distance from this to here. And from here, you have to go 1, 2, 3 to here. So that's a way to compute the distance. Well, the second method, the data diffusion, will first link everything. So I will make a link between this with every other link, other nodes. I will make a link between this with any others, because it's fully connected. Every node connected with every else. Then from here, I will select a cutoff. Say, okay, the D has to be smaller than a number, or cutoff. So if I found, okay, this distance is too far away, so I just remove it. This is too far away, I remove this. This is too far, I remove. So I apply this cutoff. I will remove a lot of those linker, which above this cutoff, then the rest of them is the network which I use to approximate this local neighborhood in the manifold. The most commonly used cutoff approach is called this Gaussian kernel, okay? So basically, Gaussian kernel is like this, okay? Distance. So that's a distance. So that's a distance. You convert this distance to basically whether they are neighbor or not. You have to be small enough, okay, in order to be highly likely to be a neighbor. If you move away, and you become least likely to be neighbor. So that's some kind of this stochastic get involved. So that's called a diffusion, just like the water diffused from a nearest to far distance. Okay, so Laplacian eigenmap. So this is a algorithm proposed in published in 2003. Okay, from this paper. So that's an algorithm description. So if you look at this, so we will first build a graph. So this is a network. You can use either the first algorithm I just proposed, KNN, so, or you can use Euclidean distance, then set up cutoff, okay? Then you can choose to use weighted or unweighted. So if you decide every linker have the connectivity strength as 1, so you put just 1 for given to every link between 2 nodes. Or if you want to have introduce some weight, so if you are relatively far away, although you're still linked, but the weight will be weaker, okay? If you are linked and also close to each other, and I will give you a higher rate. This is through this this kernel transformation, okay? Anyway, so these 2 steps will guarantee you get a network, okay? So the network will be a matrix, say a graph matrix, okay, graph. Let's see, using W, because this is a weighted. So this algorithm will compute the, so this network is a matrix, okay? Adjacent matrix, basically. So from here, you can compute the Laplacian matrix. Okay, so another. Laplacian matrix is just there's a degree minus the adjacency matrix. So if you revisit the previous slides, one of these slides should have this, how to compute the Laplacian matrix. Can't remember exactly which Laplacian matrix that's for. Can't remember which week, but the previous one. So this can be computed very easily, okay, through this first two steps. You get the network between these points, and also transformed. Only consider the neighbor, nearest neighbor as a linked, and then you get the network and you get a Laplacian matrix. These two matrices will be used in construct embedding. So W and L will be used in next step. So the next step is called generalized eigen decomposition. So remember we had eigen vector, eigenvalue definition, if you still remember. Okay, this is a squared matrix. Then if you apply squared matrix to an eigenvector, you will not change the eigenvector. So the results of this will be lambda, which is a number times eigenvector. That means you only change the magnitude of the eigenvector, but not rotate it. So that's our eigenvector definition equation. For Laplacian matrix and the last step, is something similar, okay? This is called generalized. Let's see. D. So the F here. So I replace. Let's see. L. F. So F is this. And then you have lambda. Then df. Okay, d. Okay, just change the variable name, basically. F is this V, so this is eigenvector, and eigenvalue is still the same. And L and D are all squared matrix. So this make this is like a simplified version of eigenvalue relationship with matrix A, but now it's replaced by two matrices, and this formula is called generalized eigendecomposition, eigenvalue problem. Okay. So let's revisit these first two steps. See how we can get L. L is here, so L D minus W. So we can have a D which is a degree, the O0 here, and only have values in this diagonal position here, okay? So that's degree, this is matrix. DL will be used in this formula to get eigenvectors. So in this example, we have a graph. Okay, so you can construct graph using KNN. There are different flavors of KNN. So if you do directed nearest neighborhood, or just not consider a direction, so you can have this undirected. If you require mutual KNN, so both this has to be recognized to each other. So this has to consider this as a nearest neighbor. This also has to consider this as a nearest neighbor. They have to be mutually nearest neighbor in order to keep that link. But if not, then you remove that because this will consider this as nearest neighbor, but not the other way around. So that's been removed, the link. There are different ways to do KNN. But you can make link between this, create a graph. Then you apply this one of the kernel function, okay, to convert the distance you compute into a weight, okay. This kind of transformation or some kind of soft cutoff. But right now, this is a strong cutoff, 0 or 1. But if you apply that, so you make this 1 into respect to the distance to transform the weight between linkers. Then that's Laplacian. So once you have this W matrix as a representative of the graph or network, then you can compute degree for each node and put the degree in the diagonal position, make a square matrix. The Laplacian matrix will be computed by D minus W. So this is weight matrix. So once you have these two matrices, you just apply this generalized eigen decomposition or eigenvalue computation. Then you get F. So F, you have a series eigenvectors. Those eigenvectors you can keep. Say top 2, you have a 2D embedding. Top 3, you have a top 3. You have 3 embedding. So the eigenvalues, eigenvectors will be the embedding you will get at the end. This is a small example with only 4 nodes. So 1, 2, 3, 4, then you compute the nearest neighbors, and you found this link. This is a graph, adjacency matrix. And then you have W, so this linked because 1 and 2 are linked, 1 and 3. 2 and 3 are linked. One linked with all the 3, but they are not linked to each other. So this is your so-called unweighted adjacency matrix. This is a network. So this is a degree. So first node have 3 linkers, so degree is 3, and the rest of them each have 1 link, so only have 1. So this difference between them is the Laplacian matrix L. Then you do generalized eigenvalue problem, you get F, and F will be your embedding. So that's this part is the Julia code, Julia language. But you can use MATLAB have a similar function to do this. So this is a comparison between PCA and Laplacian eigenmap. This is for linear embedding. So this is for nonlinear. So PCA not considered as a geometry aware. So you lost the intrinsic geometry of the data. So for this, if you apply PCA, you will get every points superimposed onto each other, not in respect to the structure of the of this manifold. Well, the other manifold learning algorithms, diffusion map, Laplacian eigenmap, and all this can do a much better job to basically unwrap this structure. This is a very simple example. You have very clear visualization, but when you have high D data, you don't see that, you know there's some structure there. So you have to rely on manifold learning to do this unwrapping, to open up for visualization for the downstream purpose. There are more examples of those fancy nonlinear high dimensional 3D manifold structure. And for example, here you have S shape and also this net structure. You should have to find a way to respect to this local structure to get the better embedding for the 2D. PCA definitely will fail in this case. So manifold alignment, a manifold learning approach has been utilized in single cell data analysis. So the researchers who applied this method, the most successful one is from the Yale group. Her name is Smita Krishnaswamy. Okay, her lab has pioneered quite a few of this embedding manifold learning approach applied to single cell data analysis. You see her review paper, perspective paper published in 2018. So at a very early stage, she started to use this. For example, this is a KNN network for the cells, okay? And try to kernel transformation, the diffusion, and follow the manifold structure to compute the distance, to do visualization, okay? To design the algorithm to do imputation. So this is follow manifold structure and then try to find the neighborhood of cells and try to copy and merge the expression profile from one cell to another neighbor cells. So it could be your input is very noise and sparse, but after this so-called magic algorithm, you smooth out those relationships, follow the whole population of cell structure, manifold structure as well. Then once you have this imputated matrix, a lot of this downstream statistics analysis, which rely on non-zero values, will be become more powerful, because if you have a data is sparse, there 80% of zero, only 20% of cell have those gene expression. Any correlation or regression, you just cannot apply very well for those. For example, these two gene A, B, you don't see anything because they all zeros, a lot of them. But after this magic imputation through this manifold learning and the smoothing, then the relationship between these two become clear. They are positively correlated among the cells. And it will help you to enhance the downstream analysis. Sometimes what we found is very useful. Okay, it's time for break. 5 minutes. Okay. Okay, so single cell data is inheritable. By nature, it's high dimensional, right? So that's not strange that we see manifold learning have been used in pseudo-time analysis. Last week we talked about. So in that case, we have this KNN applied in many different places. Monaco 2 utilize some of those. So you have to follow the KNN network in order to construct this trajectories, okay, move from one cell to another. This is also this idea of manifolds. Respect to the manifold structure, you cannot jump from one state direct to another of the cell, so they are not follow the biology. Manifold alignment is to have two different state sets, and you try to align them and consider both of them are manifold structure, and the structure they are nonlinear, and you want to match them to align them, just like the CCA last time we discussed. That's from a linear transformation, rotate and then try to match the embedding or projection. And here we consider these nonlinear structures. One of the examples is like this, and A and B are two different objects, but they rotate in the same way, so you want to match the location of this with this and that with that. So alignment is try to produce projects, okay, between sets. Could be more than one, or maybe more than two. You have multiple data sets, and they all has its own manifold, but you try to align them, try to match the alignable data points together by respect to their own manifold structure. And the foundation idea of this is to utilize relationship of instances with each data set to strengthen the knowledge of relationship between data sets. Okay, after you map them and align them, and you can learn from each other. The same object, you have two, basically one, for example, one cell, you have two different measurements. Even though they are not measured at the same time, you align them, then the feature you can transfer from one domain of the knowledge to another. So the illustration is something like this, and you try this structure, okay? So data one is blue until go through this structure and you get a data n at the end. And in another domain of the measurements or experiments, you have data 1 here, and then data n is here. So you try to reduce the dimension of this 2D and into this 1D order, and then they match, aligned, side by side. Another example is like a 3D protein, okay? So this blue one is much smaller in scale, but you can align them. They're not perfectly aligned. The structure, they're highly similar, okay? Then you at end, you have to align the structure. This is one of the early stage published manifold alignment paper, try to align the measurements of about a different data sets together to align this cell type. This is another publication which see, okay, this is a linear alignment of the CCA not work, and then they decide to use this nonlinear manifold alignment. This is another applications for manifold alignment to single cell data. They have samples which collected from different time points. Okay, here is gene expression. Here are the chromatin accessibility measurements. They try to align them, the samples. There's another publication using manifold alignment. This is another, okay? So why those analysis interesting? Because now we are talking about the multi-omics technique. What that means is if you only think about this bulk RNA-seq, and then at the same time, same cell, you can do epigenetics, okay? Same population of cells. They are not the same cells, but you can have a expression level measured, and for the same population, you can have the epigenetics information, the methylation, acetylation, but most commonly used are the ataxic, so you can get open chromatin structure information from them. So in that case, you will have one matrix and another matrix. So those are the cell, many different cells. They form a structure, okay, a manifold. They are not linearly related. You have some manifold structure for the cell population. And here also, for the cells you measured using different omics technique, you also have those population of cells also have a structure. Because they from the same population of cells, so you believe this manifold structure are similar to each other. They're alignable, okay? So if you can do a good job to align them, then the alignable part of this cell for the same subgroup of cells, you should have their gene expression as well as their epigenetics information shared, okay? So in that way, you can see, okay, at this particular stage of cell, you have this particular expression and the particular epigenetic signals. So you can make the cross link between signals, okay, different domain of the knowledge. They shared the same population of cells. So this is only two, and you can have methylation, you can have surface protein, you can have, I don't know whether T cell, B cell receptors information can be incorporated, but the idea is to try to align them. This is another called coupled NMF, non-negative matrix factorization. So it's slightly different strategy, but same goal, same purpose. You try to align single cell RNA-seq with single cell ataxic information cells to align them. So if you think this is a gene, this is a peak, they're ataxic peak, and those cell population, for example, this brown one can be aligned with brown cells here, and this dark orange okay, purple, can be aligned. Then you can study the gene and their peak, how this peak changes and regulate the gene expression, or other way around, whether gene have an impact on the peak presentation. So you need this alignment before you can do downstream. So now I'll talk about the single cell ataxic analysis. So single cell ataxic analysis stands for the chromatin. This is the same machine. 10x Genomics use the same controller to do this single cell ataxic. Ataxic stands for IC for transposase accessible chromatin analysis. So you know eukaryote cells have this chromatin, including euchromatin and heterochromatin. So euchromatin, and you can have this structure and can open and allow DNA to be exposed to the local environment to either to be bound by the transcription factor or other regulatory elements. Well, the heterochromatin will be compact and not related to any transcription or translation. So we want to understand which part of the genome is open, and once it's opened, it means the cell is utilize the genetic information within this DNA, these fragments, to do active transcription or other modification. So we want to know which part is open and which part is closed. And this euchromatin open is also context dependent, okay, cells at a different stage. This particular region may open or may not open. So that's involved all this chromatin remodeling. There are a lot of signals going on to control this. So open or not open also have impact on this modifications, methylation, histone modifications. If it's not open, they're all those regulation events cannot happen. So we really want to know which part chromatin is open, which part is not. So this open or not open, not directly linked with methylation or acetylation, because you have to use other antibodies to measure those signals. But it just gave you some broad idea about the openness of this region. There something may happen there. So oftentimes those are somehow linked. So this open chromatin analysis will help you to understand epigenetics in the cellular level, okay, especially if you can do this. ChIP-seq is which you get this open structure, and also you broke it up with antibody, enrich these fragments, so you know, okay, this part should be opened because you sequenced a lot. You fished out a lot of these fragments by using this antibody, so you know this binding happened somewhere here. So this is a ChIP-seq idea. So this ChIP-seq really rely on the antibodies. Normally we cannot do it at a single cell level because you use one antibody, you only measure the binding of this antibody to different regions in the genome. But we use ataxic to generalize just any place which open, I will see the signal. Maybe not related to this particular protein, particular antibody. So that's the process for the ChIP-seq analysis. It's related to ataxic, but not exactly. So ChIP-seq idea is you sequence that fragments. So because you have antibody binding to enrich these DNA fragments from the genome. So once you map this back, you know, okay, this particular binding signal are enriched in particular region. So that's a binding place. You can locate in the genome and which part this binding happened most. So you see the TF transcribing factor that binds to this. There's two peak effects, but the algorithm can correct this two peak signal and find the exact location where the binding happened. Those are the files which show you the peaks. Okay, those peaks are from a ChIP-seq or from a tag-seq. So it depends on which antibody you used to represent which transcribing factor. So you have different peaks. So for example, you have the binding for this brown transcribing factor in this location, but not a blue one. So that's in the beginning of this particular gene. So those information are very useful to help you to construct using this ChIP-seq information, particular protein you designed for measure its binding affinity in the different location of the genome. A tag-seq is more generalized. It's just cut this open region. It doesn't matter which transcription factor located here. Is a TF1 or TF2, it's always have an open structure. Then you can measure it using a tag-seq. You see this region is opened, so you start to see the peak measured using a tag-seq. So a tag-seq using this transposis, which specifically cut those open regions of open chromatins. So once you cut, you generate a lot of these fragments. These fragments will be mapped back the place where you cut more, and you get more fragments. So 10x Genomics designed this IC and using bulk RNA-seq idea, but do this reaction in the droplet. So that's why you can get nuclears. Each nuclear is one cell, okay? So the single nuclear, basically. So you measure these nuclears or individual cells, you get the peak information, just like you get gene expression, but those peaks will not be named, not like a gene. It's just a location, the chromosome 1 position. So you get the same structure. It's a matrix of the data, but you are not getting any gene name. You only get the location of this peak. But you can use this peak to characterize each nucleus. If you have two nucleus have a similar peak profiles, they will be grouped together, and you can use same t-SNE or UMAP method to do this. Embedding, you can also cluster this nucleus depends on the embedding, and by figure out their peaks and where they located, because peaks sometimes associated with the active gene expression. You can annotate those cells using the location of the peaks and then figure out the cell type, especially if you know what cell type in your sample, okay? Any questions for this? Okay. So this is illustration of the relationship between bulk ataxic and single cell ataxic. So if you look at this matrix, there are peak locations, okay? And then each row is a single cell or single nuclear. And if you have a peak, you get a darker signal. If there's no peak, you get empty space here. So here is the several thousands of cells, and if you add them up, and you reconstruct the peak, which you get from a bulk ataxic almost perfectly. So bulk RNA ataxic, it's just aggregation of individual cells, signals. But now we have this access to single cell level ataxic peaks. For this resolution, you will be able to find the place where every individual cells have a similar peak. Okay, something like this is a constant base. Basically, that means this peak appeared in all the cells. And if you accumulate those signals together, you should have a higher, strong peak in the bulk ataxic. Well, someplace like this variable peak, just like highly variable genes, is expressed in a few cells, but maybe not expressed in many other cells. So you have a very sparse signal. So this is a place which this peak change its representation in the cell or not, variably. And when you accumulate this, you get a lower magnitude of this peak height. I don't know anyone study the highly variable gene using these variable peaks. I don't believe anyone is using this. But if you believe that's important, because this peak could be very important to actively regulate something. So the highly variable peak analysis may be a new analysis. We haven't done this, but we could do this. And also where this peak is located is important. So in general, we believe the peaks are regulated in upstream of the gene under its regulation. So because the open chromatin have gave the accessibility to a lot of bindings of transcription factors or enhancers in that region. So if this peak is in front of this upstream of this particular gene, we normally believe that's where the place of regulated elements regulates expression of downstream genes. Okay, so before the joint kits become available, we have to do this single cell ataxic and single cell RNA-seq separately. So although you have the same population of cells, you have separated the cells into two populations. One is for doing this ataxic, one do RNA-seq, one do ataxic. And then you have a problem to get cells to be aligned. Okay, you have to find the cell which with the gene expression profile and the cell with ataxic profile. They are highly similar, and you put them together. So you have to do this alignment. So that's where the manifold alignment get involved. People start to develop those methods, including this. I just introduced the coupled non-active matrix factorization. The purpose is to align cells to find the similar cells together. So you can use gene expression to explain the peak. You can use the peak ataxic information to explain the gene expression. If there's if you shuffled and randomly align them, then you will get messed up the signals. So that's what talking about. But now But now all this statistical data science algorithm become useless suddenly because the technique already allow you to measure the single cell ataxic and the gene expression in the same cell. Okay, so you don't have to use data science to align them. So manifold alignment is not useful because they already know you sampled them. They are technically aligned.
So in the single cell multi-ohm, instead of having two separate samples, we can use the same sample and it will give us the attack and the gene expression data. Is that what?
What I described is for the same sample, you have to see 100 cells, you have to separate 50 to this, 50 to that, to do that. But now you can have 100 cells to do attack and expression at the same time. Yeah, so that's a difference. It was, certainly, we always deal with the same sample, but the same population of cell, but you have to separate them, the process separately, but now you can do on the exactly same cell, these two measurements. So you don't have to align them anymore. So this is called single cell multi-omics, single model. So, so in your description of your method, you have to specifically mention if you do this multi-omics, you have to mention, I did a single cell RNA-seq and ataxic from the same cell rather than do this separately, align them statistically different. So we can do ataxic plus single cell RNA-seq, but there are others. We cannot do methylation at the same time. We cannot do, I don't know if you can sequence the DNA and mRNA at the same time. We cannot do the proteomics now. So there certainly it's not completely alignable. You have to do align if you do something else. It's not completely achievable to have a real multi-omics for the same cell. You have, this depends on the same population of cells, not the exact same cell, but ataxic and RNA-seq is okay now. So assume you have the ataxic and RNA-seq from the same cell. The pipeline I would suggest is using this extension of the Surrey package, SIGNAC. Okay, so they specifically develop this package which can help you to analyze single cell ataxic. They have some extension of function. I don't know whether current version is number 5, right? Surrey. So from the version 3, they have an extension to do this ataxic single cell RNA-seq integration analysis. So we don't have this function in our toolbox, so you have to rely on R if you want to do single cell ataxic analysis. Tensor, okay. So tensor is a 3D array, okay, or even higher dimension. So you have a qubit of the numbers rather than the matrix. You can have one matrix plus another multiple layers. So just like this, and from 2D becomes 3D and give you more structure, high dimensionality, more information from one data set, structured data set. But of course, computationally more difficult. So 1D vector and the 2D matrix, 3D or third order tensor. You can have a higher, okay? That's a representation. we actually have a lot of tensor data sets. If you have a colored picture, they have three primary colors. So this is the three layers of the matrix they plus together and to form this colored picture. So you have three channels. So this single data structure is a tensor. If you have a picture, so each picture is a tensor 3D, but because of the time theory, so you have actually a 2D, a 4D, so fourth order tensor data set. There are many other examples. Here is a recommender system. So there are different time points. So physics use a lot of this general relativity is a function. All these are the tensors variables. So it's a computational more challenging computer if you have a implementation or algorithm can handle this, then you can write data x, y, z and equals what. For example, this one, you don't have this particular structure. Data not allowed to have 1, 2, 3 dimension, x, y, z. Then you have to write in a vector. Okay, in this case, this implementation, I believe it's in the C++. And then there's no tensor structure defined. So the data, you have to use a vector, okay, a very long 1D string, like array to represent the three layers. So to code, to program using this, that without a tensor definition will be more difficult to handle. So fortunately, a lot of high-level language, Python, R, and MATLAB, of course, should be able to handle this tensor structure. Vocabulary, and in the computer science, people talk about number array, 2D array. Mathematics may call scalar, vector, matrix, but they are the same, right? And the array and the tensor. So tensor can be useful sometimes to do, if you do a tensor decomposition, to learn the components in the data. If you have a high dimensional data, it's not a spreadsheet, but it's a layer, different layers of the spreadsheet. You analyze them together rather than deal with this spreadsheet and that spreadsheet. It could be January, February, March, and you have different tables. But if you concatenate them together and you have a 3D and put all this into the data analysis using tensor decomposition, you can learn more. And so that's the idea for to do this tensor decomposition. So on your left side is the matrix decomposition. So you should be familiar with this by now. If you're not, revisit this components and decomponent lecture several weeks ago. So the idea is to decompose your observed matrix A into the product of the several small matrices, okay? This decomposition into the product of these two. Same idea, if you have a high 3D, you want to decompose into three matrices. Okay, three matrices multiplication will reconstruct the whole thing. So you want to learn the structure. You want to know how I can decompose this matrix into those combinations of this vector or matrices multiplication. Same idea is here, just high dimensional. Like 3D, you need to have 3 components multiplication. So there's a matrix multiplication to reconstruct tensor. So canonical decomposition, they're historically have different names, and they all called CP. So the CP is one of the most important and commonly used decomposition, tensor decomposition algorithm. It's a kind of optimization problem, so you try to find those vectors, or the multiple vectors, their multiplication together will construct the tensor. And they can also ranked, ordered by this contribute more, and this contribute the second, and then you can have more less contribution. And then you can have, you can see, okay, I only use top K components to reconstruct this, and a kind of approximation of the original data structure of the tensor. So the CP method is using so-called alternating least squared. Basically, you fix one dimension and then start to minimize the rest. And then from ABC, so you have ABC 3 components. Then you fix one, try to minimize, fix the rest. So you take a turn. So you do this A dimension first, and B dimension, the C dimension. So just take a turn and to approximate to reach the combinations of the values of those vectors or components in order to minimize the difference between your dot product, reconstruct tensor is here, and with the real data. So you try to minimize this difference. So those are some details in the least squared alternating least squared algorithms. To make it faster, there are some kind of randomization acceleration approach. So that's a decomposition, and also use a CP low rank approximation. So you try to keep the most important ones and get rid of the least important ones in order to do this approximation, and sometimes you denoise the original data. Another decomposition method is called Tucker low rank decomposition. So this have 3 fibers, they call. So those put together to reconstruct the original. Applications, you can have analysis for this time series change of the communication network. So this application in the a social science, basically they have a school student network, and also they capture this their communication record across the time. So you have a tensor structure to learn their communication patterns. And in this way, in this analysis is a mouse study to study the behavior of mouse and their neuron activation. So also this is a multiple neurons, and at the time they follow the signal of neuron produced, and there are different trials and at different time. So you create this tensor and start to decompose and learn different components. I encourage you to watch this video and to get the story, and the different components may mean different things for the different parts of neurons activation. It's very interesting. Part of this could be the rewarding and gave you when the animal finished the experiment, gave it the food, and then some components of this neuron activity can be learned. So this is one of our applications. You probably have seen this. So in this part, we have a tensor structure, and from multiple networks. Then we did a decomposition, and then threw away those less important components, and then use the most important ones to reconstruct the network and take average. So this tensor average and gave you a denoised version of the network. Okay, so this improved the significance of the accuracy. We found this useful. So at the very end, I want very quickly mention the velocity, because I promised to explain this a little bit from last week, but we didn't have time. We still have 5 minutes, okay? So this is a very different technique, and the people try to learn using the splicing patterns from a single cell data to learn the change of activation. So if you know the mRNA, you will start from a produce pre-mRNA, then you go through this splicing events to cut and to become a mature mRNA. So if you produce pre-mRNA and your mRNA processing in balance, the same rate, then you expect to have a same unspliced and with spliced mRNA at the same concentration. So that if you detect this information, that means this particular gene you get from individual cells, this gene is being actively produced and processed at the same rate. So this is a kind of a balanced, steady processing. But if you have a gene has been downregulated, that means you stop the producing of pre or native mRNAs. So relatively, you have less pre-mRNAs compared to matured mRNAs. If you observe a gene which in this pattern, that means this gene start to be downregulated. So you produce less pre-mRNAs, and you have relatively more matured mRNAs spliced. So this imbalance to tell you whether this gene has been shut down at that given time. So next time you should see less pre-mRNAs. So in contrast, if you have a gene is under upregulation, so you start to produce more pre-mRNAs with unspliced compared to spliced mature mRNA. So the rate of the producing is increased, so you get more pre-mRNAs. So based on this balance, you can put the genes in different regulation phase. Okay, you have more spliced, less unspliced, that's under downregulation, and you can put this look at the gene and this to put the cells in the different states. So the most convincing example is the paper which introduced this method using circadian rhythm genes, because for circadian rhythm genes, we have a sample at a different time, and we know some of the genes will be upregulated, downregulated based on the time. So we have some ground truth. And for example, this CBS genes, we know at 6 a.m. is start to down-regulate. So if you look at this balance between spliced and unspliced, you see a lot of spliced compared to unspliced. That means this gene at 6 a.m. is being downregulated. In contrast, in the afternoon and 6 p.m., this gene has been upregulated. And if you look at the relative abundance of spliced and unspliced, this location the samples become here for that particular gene. So it's very clear, and using this balance between spliced and unspliced counts, you can infer a gene's activation by the rate. So if you incorporate multiple genes together, you can start to explain whether the cell is going to that particular stage or not going to upregulation, downregulation to some states. So that's the idea from the RNA velocity. In theory, it's a very powerful approach, but because of the quality of data, the counts may be not as accurate as expected, and I don't know how to incorporate different genes together. But there are some criticisms of this method. Not numerically very stable, okay? But the idea is good. So, to look at splicing and unsplicing relative abundancy in order to infer the up and the down regulation of a particular gene's expression. Okay, I think that's all for this week. So, there. Okay, so see you Thursday in the computer lab.